Hi, everybody. Thanks so much for joining. Just letting folks kind of trickle in here. We're expecting a few more folks, so uh, just give it a couple of minutes and then we'll take it away. But welcome to our Team Science and Translational Medicine talk today. And we're really excited to kind of get your perspectives and feedback as we kind of close. So um, give it a couple more minutes and then we'll start. All right, let's go right ahead. And then as folks, I've got some promo stuff to review. So thanks everybody. Good afternoon. I'm Grant Wells, Director of Innovation and Development for MCHRI here at Stanford. Today I'll be joined by Juliet Knowles, who is an Assistant Professor of Neurology and Pediatrics here as well. She'll be talking kind of about her project that relates to our Team Science and Translational Medicine program. And I'll kind of be doing some overview and background, as well as kind of describing the program in a little bit more detail. But first, wanted to get into just a thanks to our education committee who helps put these seminars on. We're really blessed to have their support and engagement as we kind of come up with different topics and different programs, including our large symposium in October every year. Just a quick note, our next seminar will feature Tom Robinson and Katrine Svensson talking about kind of the translational spectrum of obesity, reviewing things from a pharmacologic background to fully implementing them in programs. And I think it's a really great timed discussion based on our talk today to kind of flow to that. So really wonderful opportunity given all the exciting innovation that's kind of happening in the space. A quick plug, just with some events that we've got going on. We have this immunology of autoimmunity and antigen presentation, which is in honor of the late Betsy Mellons, who was one of our pediatric faculty here, really a great mentor and specialist in those two areas. You can see we have a really wonderful cadre of presenters from the NIH and other notables around the country. Please scan that QR code if you're interested in signing up. Also, if you go to our website, the registration link's on there as well. It'll be a great time. The event will be hosted over in CAM on May 1st. So don't miss out. I think it'll be a really lovely presentation and great opportunity for some collaboration focused on this space. Just another note, just save the date, June 5th. Coming up, we'll have another Stanford Medicine Children's Health research event. Just put that on your calendar. Really wanted to take a moment actually here. If you know folks who you want to nominate in your work as a research champion, just based on their engagement or the support, kind of wherever they fall, whether they're at the hospital or here at the academic medical center, just take a moment and scan that QR code and kind of nominate them. Hope you can kind of recognize them for all their wonderful efforts, as we're just so blessed to have such a wonderful community that support all the given research areas that we work on here on campus. But just without further ado, really wanted to dive into this seminar today. And feel free to ask questions. We'll try and get to those kind of at the end. You can use the chat feature to do so. But really the purpose of today is how we prioritize diverse collaboration and how we think about accelerating innovation. So just kind of in broad strokes, one of the things I wanted to focus on is just why is team science necessary in translational research? I think this question we think a lot about. Maybe we hear it sounds very buzzworthy. Um, it's topics of funding announcements. We're putting it into different grants that we write, but really at its core, this is a very different method of management and engagement within our research that can really produce some really nice fruit. So, um, you know, as we just kind of think about that, when you're on your own, there's so many different avenues to take. There's so many different processes and areas of investigation which you can pursue. And going it alone, there can be certain advantages to this for sure. They can you can get to places quicker. You can work on things in a more dedicated way. But in reality, um, others, when we interact with them, can bring up different aspects of a line of research that may eventually be pitfalls down the road, and having them engaged much earlier um, is really something we wanted to focus on as an institute, but also within our team science and translational medicine program, is just finding the right collaborators and establishing what we're kind of calling verticals earlier. And we'll get into that in a little bit. But why I really wanted to focus on this is from a very logistical point of view, is that as we consider this valley of death, we typically think about this only from a preclinical to clinical aspect. But there are iterative steps that we come across, which I think this diagram describes really beautifully, and take no credit of my own for creating it. But it's one that, as you can see, having different perspectives engaged, either in the basic sciences or the clinical trials related, or even in the translation to practice and as well as the community, are really vital. There are critical processes, and I've gone through a few case studies myself where when when you're not thinking about all the outcomes that could possibly occur, you may skip steps or make assumptions which can be pretty detrimental. And having let's say a community engaged researcher, or even somebody from the community engaged very early on in your translational endeavor, will provide dramatic support over the lifetime of your project. Not only just from the difference of perspective, but you'll have a partner when you reach that phase, for somebody who can help you think through different aspects that can come up. And gotta say, I think Tom Robinson really has done that really well, as many others here on campus have. We'll kind of hear Juliet's story in just a minute, but really taking this to heart is something that can really be a positive to our lines of research. So when we're thinking about team science, what are our critical key priorities as we have these lines of investigation? And one of the things I can't think of enough when we think about diversity, not only diversity in background, but also diversity in role. So having a clinical researcher engaged with a basic scientist, as well as an expert in community engagement and population health, really can help you start building that case, and having those members on your team, and really facilitating a management structure that allows you to have open conversation in a structured way, where you're thinking about a project much more in a broad sense, can help you identify areas where we may need to focus more on the patient and the overall outcome, but also utilizing effective tools and methods for that collaboration. So, making sure you have shared workspaces where it's transparent and easy to for others to kind of engage. You have documented processes, either for authorship or what a collaborator is and how they function, when your meetings are, just simply so folks know when and where they need to be. All of this, I think, is critical as we look at the end, just what the overall goal is, which is accelerating research at each phase. As kind of going back to the the previous slide, we don't really want to go back in our research. We want to go forward. And so how we do that, I think, is an iterative process, but involving our colleagues sooner ultimately ends up in the possibility of us avoiding some of those pitfalls that may occur, that may stumble us if we're going at that alone. And so what does this mean here at Stanford? Well, this is just, I love this slide. It's one where we're just trying to show the breadth and depth of what we have and how they all interact here on campus. And there's more activities on our campus here at Stanford than even what are posted here on this slide. And we're getting more every day. But these are more opportunities for us to identify collaborators at different centers, leverage different skill sets, build our teams effectively with new and different tools and resources, or even furthering our own education of what it means. But these are just some quick things to kind of take note as we're thinking about building our teams and designing for translational research. And so I think this slide says it best. Our overall goal for our team science and translational medicine program is we want to establish these verticals. Those verticals can really be, in any given area. We want to identify ones that obviously make an impact to maternal and child health, but we want these projects or verticals to really focus and have a trained translational faculty member at its core. And why that's so critical is because that to us is the leverage point of innovation. Having someone who can bridge the gap between the bench and the bedside as the key catalyst for the research project is really a great engagement factor. But also making sure we have representation from our public health and innovation and commercialization colleagues, and finding new opportunities for clinical researchers and as well as our trainees. What we ultimately hope this looks like over the long term is we're increasing our external funding. We're establishing our preeminence in given areas via clinical excellence, meaning we have access to new therapies or different tools that other institutions may not have. But also it's a focus point for our philanthropic efforts as well. These are areas of engagement that we can have with our development colleagues to think about what's new and what's innovative and how are we improving care overall. So a quick transition. This is our lovely past cohort of TSTM colleagues. They're currently in the midst of all their projects, and I think really doing some wonderful work. You're going to hear about it with Juliet's project here today. But we were able to take them about a year ago to Silmar, and they were able to engage kind of in a structured setting with some great mentorship and training materials to kind of engage in their individual verticals and make some headway. I think for program objectives, just if you're interested in considering this program for your own research area, these are kind of the quick areas. We want to have interdisciplinary teams. We want to help you build those teams. If you're saying, hey, I really want to connect with a given colleague, in the Woods Institute or, in another medical department, that's something where we would love to facilitate that interaction. And feel free to follow up with us after the seminar. We're definitely available for that. We want to have translational experience, in a connected fashion, very structured setting as we're kind of learning those key priorities of translational medicine. We want to have that done in a way where it's well defined. And what that does is that ultimately you have a programmatic roadmap for how you're going to execute your project over the lifetime and how it's going to build upon itself. And then ultimately, like with all things, just helping folks apply for extramural grants. And we have wonderful colleagues that are on this call today who are experts in that, and we partner there. But but also we're always looking for new and engaged opportunities as new opportunities through the NIH and other entities come out. And then what are our work products? I think this is one that's really important to, as we define what success of this program looks like. And so we want to engage with mothers and children who have really challenging conditions and foster an environment where new novel therapies or policies will ultimately lead to improvement in their overall quality of life. That at its core is relative to the mission of what MCHRI does as a whole, but also is something as we're looking at a faculty development program. This is something where we want this to be at the forefront of the program as well. We really want to have this focus on preeminence. We want to have the visibility and the recognition for our translational and clinical research. I've kind of gone over this. This is hard work, but the rewards are really incredible when we're able to see things that we've worked on in our labs and then see them applied into our clinics as well as into our community. So that's something that, as a secondary outcome, is something that we're really focused on as well. And then at its core, we do need funding, now more than ever, I think, just to focus on, different lines of revenue generation, and revenue meaning not from a profit perspective, but, to drive our research programs forward. We want to open up potentially new areas of funding for our endeavors. So that is something that, within a package, it's a longer term goal, but is something that also we're focused on. So these we feel are major products of how we're going to define success and ultimately wrap our heads around as we kind of embrace our research community on this TSTM topic. Just here's up from that cohort where I showed you the photo at Solmar just a bit ago. Here are our project teams, and you can just see kind of the really interesting aspects of what folks are doing. We're really blessed to have such a wonderful collaboration around 22Q with Katya and others. We have some really cool stuff happening in inflammatory bowel disease, but also look at what Stephanie Chow's doing with the pledge campaign related to firearm safety. But really wanted to take time today and engage with Juliet Knowles, who has this wonderful lab that she'll tell you about, but also has had some recent success in engaging with clinical partners around how we're treating synaptopathies, and really kind of designing a clinic for those children that are impacted by these diseases. So want to take the next step and just hand it over to her. And just thanks so much for your time.
Great. Thank you so much, Grant. That was a really nice overview of this program. And thank you so much for the opportunity to come share our work today. Share my slides here. Okay. Are you seeing slides in a presenter view or slides in a presentation?
Presentation view, sorry.
Normal slides.
Right.
Okay.
Yes, normal slides.
Excellent. Okay, great. Yes, so I'm going to be telling you a little bit about our experience with the TSTM program, which led to a project that we call Precision Medicine for Pediatric Synaptopathies, a team science approach. And as Grant mentioned, I am an assistant professor here. My lab is about 3.5 years old, so I have a basic and translational research lab focused on pediatric epilepsy. And then as a clinician, I practice as a pediatric neurologist specializing in epilepsy. So first, I'll tell you kind of like what it was like for our team going through the MCHRI TSTM program, starting with the retreat in February. And then I'll give you some exciting updates on our progress 1 year out. So if we rewind to about a year ago, this is kind of where we were. So one of my colleagues in child neurology, Chris Lee Messer, and I had just launched this new clinic that we call the California Synaptopathy Clinic. And so you might be wondering, what is a synaptopathy and why would you want to have a clinic focused on that? So I'll tell you a little bit about this. In child neurology, we frequently see children that have epilepsy, autism, intellectual disability, or a combination of these factors. And with the increasing availability of genetic testing, we're realizing that a common cause of all of these things is disruptions to genes that govern synaptic function, such as SYNGAP1 and STXBP1. And I'll be talking about both of these diseases today. But together, these disruptions to these genes are referred to as the synaptopathies. And just as an example of how these disorders tend to manifest clinically, I'll talk a little bit about SYNGAP1. So SYNGAP1 related disease results from loss of function variants in one copy of the gene that lead to haploinsufficiency. And SYNGAP1 encodes a protein that is required for normal synaptic development and function. And haploinsufficiency of this gene leads to a pretty severe constellation of neurological and other manifestations, including highly penetrant, severe childhood onset epilepsy, neurodevelopmental delay, significant behavioral abnormalities that some families feel is the most prominent concern, sleep disruption, autism, and movement disorders like ataxia. And similar issues come up for other types of synaptopathies like STXBP1, for example. And so the motivation to start this clinic focused on the synaptopathies really derived from this very difficult constellation of issues that these children and their families are dealing with. We had the founders of patient advocacy groups coming to us, myself and Dr. Lee Messer, saying that there was no place on the West Coast for these families to receive specialty care and the kind of complex multidisciplinary care that they really required. And so this was the motivation for starting the synaptopathy clinic. And, as I mentioned before, I have a basic and translational research lab focused on pediatric epilepsy. And in fact, one of our mouse models that we use to study pediatric epilepsy are mice that have SYNGAP1 haploinsufficiency and epilepsy. And so it seemed like we had the puzzle pieces to start building a translational research program. We had a clinic that we had just started. We had some basic work going on in the lab, but we just didn't quite have sort of the bandwidth and the knowledge to put these pieces together and to integrate Stanford resources to build a translational program. And so that's why we applied to the TSTM program. And so our project was launched at this retreat that Grant mentioned in Asilomar about a year ago. And this was our original synaptopathy team. So that's Dr. Lee Messer, that's me. This is Dr. Azeen Amini, who's a postdoc in my lab. And then we also recruited Dr. Rebecca Levy to our team. She's a clinician scientist who is a neurogenomics specialist. We also wanted to have somebody with lived experience. So we invited Mike Graglia, who's the founder of the SYNGAP Research Fund, which is a patient advocacy group for SYNGAP-related epilepsy. And then Sergio Pasca was kind to be an advisor on this project. So as Grant mentioned, this is kind of how the retreat looked. We had some didactic sessions focused on the meaning of team science. We had some wonderful examples of successful team science for translational medicine that were done at Stanford. We had a great orientation to some of the key Stanford resources that we could use to build this program. And we even heard from people at NIH about RM1 and P grants. We had interactive sessions with MCHR staff, Dr. Ruth O'Hara, NIH staff that are, program officers that could speak to sort of what's required for these big RM1 grants. And perhaps what was most invaluable was chatting with the other teams that were at this meeting. In our case, it helped us to figure out how we could start biobanking from our synaptopathy clinic. And perhaps one of the greatest things about the retreat was the uninterrupted time that we had to think and talk together and write, and to integrate all these things we were learning into a plan and a pitch. And so the goal, for this retreat was to have a 1 to 2 page pitch to start a TSTM program. And what came out of this meeting for our group was an idea about precision medicine for these synaptopathies. And specifically, Mike Graglia, the patient advocate that I had mentioned previously, his organization had been working with a Silicon Valley company called Transcriptive Bio. And this company had screened 1,500 FDA-approved drugs in human iPSC-derived pyramidal neurons and identified a subset that induced upregulation of SYNGAP expression, because remember, this is a disease caused by haploinsufficiency. And one of the compounds in that subset was nortriptyline. And this is very exciting because this is an FDA-approved compound that is still used in children. So this is a tricyclic antidepressant that was FDA approved in the 1960s for major depression, and now it's sort of a second-line therapy after SSRIs. It's still used for depression and other indications off-label. But one reason why it's not first line now is it does have some side effects that can limit its use. And this is the typical dose for children when it is used in children. Another thing that we had to think about is that there's this kind of conflict in the field about the relationship between tricyclic antidepressants and seizures. There are not great studies. There's just small studies and case reports, some reporting that tricyclics and other antidepressants can trigger seizures, and other studies claiming anticonvulsant effects of TCA, so not well-controlled studies. So, we have this promising in vitro data in the cell-based screen, but there are these questions about whether this is really clinically tractable. So this was really a perfect, scenario for a preclinical study in a mouse model. And as I mentioned, my lab actually has these SYNGAP1 haploinsufficient mice. So these mice have a point mutation that leads to a premature stop codon. And we have shown in our lab using 24-hour video EEG that these mice have progressive generalized epilepsy similar to children with SYNGAP1. And so the pitch that we came up with after this meeting, started here. We thought, okay, we'll take this drug candidate and we'll test its efficacy in SYNGAP1 haploinsufficient mice. And we then, proposed that we were going to use the pilot data that we generated with the seed funding from MCHRI to apply for larger grants and ultimately NIH funding. But I think thanks to the sort of environment of this retreat, our vision actually became much broader. We realized that this study could be a prototype that we could replicate to develop precision therapies for other types of genetic epilepsy here at Stanford, and that we could maybe even sort of make this, operationalize this into something like a program or something that we could apply for larger funding for, like an RM1 or P grant. We also, through our discussions and presentations at this meeting, realized that we needed to do some foundation building to accomplish this very broad vision, including building up our synaptopathy clinic so that we could establish a national reputation and expertise, biobanking, and that we could provide the care that our patients are asking for and that they need, which is multidisciplinary care. We realized that we needed to build an interdisciplinary team at Stanford and potentially collaborate with industry. And we needed, because this is a rare disease, if we're going to do translational work in this space, we needed to build a national network of research collaborators. So, that was our launch, and now I'll tell you about where things stand now. So after we left the retreat, we were really energized, and Dr. Amini, who was on that team at the retreat, came back to the lab and started doing the work. So she took these SYNGAP1 mice and began treating them with nortriptyline, and she set up an experiment where the mice are getting nortriptyline at a clinically relevant dose every day for 30 days. And she looked at EEG before and after treatment, as well as behavioral testing, and then collected brain tissue to evaluate SYNGAP1 expression. And the way we set this up, we started the nortriptyline at a later time point. So the mice are already starting to have progressive seizures here. So this was a reversal trial, and we set it up this way in anticipation of the conditions we were likely to face if we were to start a clinical trial. But I think, it would also be a good idea to start testing the drug earlier to see if we can prevent seizure onset in these mice. So she completed a first pilot study that I'll show you now, where she treated 7 to 8 mice in each of these groups, vehicle or nortriptyline, and wild type or heterozygous mice. And she found really a promising trend toward decreasing seizure burden in the mice that received nortriptyline compared to vehicle. She also looked at the mice in a behavioral test called the social interaction test. And in this particular behavioral test, the mice are placed in this central chamber, and then they have the option to either explore this chamber where there is a mouse that our test mouse has never met before, or an empty chamber without any mouse. And normal, healthy mice will choose to spend more time investigating this novel mouse that they've never met. And it's thought that deficits in this test are sort of a readout of autistic phenotypes. And so what we found is that compared to normal wild type mice, our vehicle treated SYNGAP mice had deficits. They did not prefer to spend time with, this mouse. And these deficits were reversed and normalized in the nortriptyline treated mice. So we were really excited about these findings and wanted to determine, start to look into the biological basis of this. And so we took brain extract from our treated mice, and we found that similarly to what's been reported in the literature, these mice have a decrease by nearly half of the amount of SYNGAP1 protein in the brain. And this is normalized by treatment with nortriptyline. So we were incredibly excited to see these in vivo correlates of the iPSC drug screen that I showed you earlier. And I should mention that this last bit of work was done in collaboration with Frank Longo's lab and Tao Yang, who's a research scientist in his lab. So we're really excited about the results of this pilot study. There is a replication study already ongoing in the lab, and we're looking at some other secondary endpoints. We're using this pilot study data to write the grants that we had talked about in our pitch. And as I mentioned earlier, we're also thinking about optimizing nortriptyline treatment in prevention versus reversal trial designs. And we've initiated a formal collaboration with Transcriptibio so that we can work together to connect findings from their iPSC screens to mouse and human data. And then Dr. Chrisley Messer, who was on that original TSTM team, is planning a potential clinical trial for nortriptyline in SYNGAP patients. And we've made exciting other progress too outside of the lab. Having this synaptopathy clinic sort of put us on the map nationally. And we were invited by the STXBP1 Foundation to become a natural history study site. And we've now been invited as well by the SYNGAP1 research fund to be a site for their forthcoming natural history study. And this was great. I mean, it brought in funding to support our, clinical research coordinators and helped us further enrich our clinic here. And this is what a typical visit looks like for one of our natural history study patients. So they come in for 2 days and they spend a lot of time with neuropsych, PT, OT, a neurologist, a genetic counselor. They get an EEG and lab tests. And one of the reasons that this was such an advantageous development for us is that we have this natural history study at the clinic, because many of the evaluations that occur as part of the study would be considered standard of care. And so we are able, through this mechanism, to provide our patients with the multidisciplinary care that they so need. And so 1 year out, we're here, and I think we're making really great headway on this work. We have generated compelling preliminary data. We're working on grants. We're planning the next steps. And we've made a lot of progress on building these foundations as well, in terms of building up the clinic, building up our national network of research collaborators, including the other sites in the natural history study, such as CHOP in Colorado, Cornell, and others. And we've certainly built up our interdisciplinary research team at Stanford. And so this was the team back in 2024, a year ago, and this is the team now. I almost can't fit them all on the slide, but we have, our clinical providers throughout Stanford, additional members of our Stanford research team, patient advocacy leaders, basic scientists, our national network, and our industry partners. And I don't think that we could have built this team without Grant's help and without the MCHRI. I constantly email Grant with questions, logistical questions about how to move things forward. And he's also given Dr. Lee Messer a lot of advice on planning this clinical trial. And if you're interested in reading more about this, our work was just featured in the Stanford Medicine magazine, and I think it's online today. So you can read more about our work on seizures and SYNGAP and precision therapies there. And, I think that engaging with TSTM has definitely moved us forward to, our ultimate long-term vision, which is team science-driven precision medicine for the synaptopathies and other forms of genetic epilepsy at Stanford. And so once again, I just want to thank our key collaborators and our funding sources, but especially MCHRI for just having the vision for this TSTM. I think it really does fulfill a great need, and it was extremely helpful to us and our group, which hopefully I've convinced you today. And that's all.
Thanks so much, Juliet. That was awesome. And it's just wonderful to see all the progress that you've made. I think now is one of our favorite times of each seminar that we have. Just love to open it up to questions and kind of get feedback and perspective. So if folks want to come off mute or feel free to use the chat. So I think for me, just to ask you a question right off the bat, just to kind of get us going. Before TSTM, what was it like navigating, kind of the resources here at Stanford? How did you find that? And then kind of after TSTM, have there been any big changes?
Yeah, absolutely. I mean, as I mentioned, I kind of had these two beginnings of ideas going. There was the clinic that we had just launched and the lab, but I didn't really know how to make the two kind of meet. And thanks to my PhD training, I knew how to do a preclinical drug study. but I had no idea really what to do after that. And not even like really who I should talk to at Stanford about that. And at the TSTM retreat, there was one day I remember where there were sort of stations of sort of helpful individuals from all, some of whom were part of MCHRI and some outside of MCHRI, that could talk to us about things like launching a website for our synaptopathy clinic. And our website will actually go live very soon. So that was quite helpful. How to submit an IND application to the FDA, how to get a clinical trial started. How do you think about funding something like that? Biobanking. How do you navigate biobanking? Who do you even talk to? Now we have an IRB approved to do that, and we're ready to start biobanking, and we've set it all up with the Stanford Biobank. So just all of those practical things that nobody teaches you about when you're learning sort of basic science. That's what became accessible to us, and just also contacts around the university. So if I don't know exactly the right person, then I have a group of people through MCHRI and other organizations and offices throughout Stanford that I now know that I can reach out to point me in the right direction.
Thanks. That's really awesome to hear.
Yeah.
Any other questions? Folks want to come off mute? Love to get some different perspectives based on folks who are on this call of kind of where they're coming from. Looks just from the folks that are on kind of this Hollywood Squares, folks from across the institution. So would love to kind of hear from folks.
Juliet, this is Sushma Reddy from Pediatric Cardiology. I'm wondering how you navigate bringing patients in to obtain samples and subsequently working with X facility to actually bank the samples.
Yeah. So we're still, smoothing out this process, but we finally have all the pieces in place. So I learned a lot about this from talking with one of the other teams, Casey Gifford. So she had, she's, if I understand correctly, she basically studies, she uses iPSC models and builds cardiac organoids essentially. And they're patient derived, which is super cool. So I talked to her quite a bit, and there is a Stanford biobank, and they have a whole procedure for obtaining a blood sample or a skin biopsy in a Stanford lab. If it's a blood sample, then it can just be collected like routinely through a Stanford lab. If it's a skin biopsy, then the way we've done it is we have somebody on the team who's trained to do that. And then there's sort of different schedules that you can kind of pick from their menu of how many samples you want stored and how you want them stored. But they, all the infrastructure for securely storing these samples and keeping track of them over time is there, and you just sort of pay a fee for having the samples stored. And so that's what we've done. And we had to obviously get IRB approval to do this, but in the end, it was not nearly as hard as it seemed.
So we've collected almost 2,000 blood samples from children with congenital heart disease. So in case any of those children have synaptopathies, please reach out to me. I'll check my biobank to see if any of those samples and any of those patients are the same, and we're happy to share samples with you.
Oh, wow, that's amazing. Yeah, and are you using the Stanford Biobank, or how do you store them?
I haven't had very good luck using CTRP or the Stanford Biobank, so I do it all myself with my team.
Got it.
I think that speaks to the importance of biobanking as well, and kind of establishing the sample collection parameters, you know, in a well-defined way. I mean, it's impressive what Sushma's doing. Um, you know, I think it'd be hard for us all to do that on our own.
Yeah.
Which you know, I did not know that, Sushma, so I'm going to follow up with you after. But I think that's something where, you know, we want to be connected and helping different researchers with some of these areas, so that they're well defined and we can use them and folks know about them. Which I think that's probably the hardest thing here on campus, is just all this really innovative work that people are doing, and you know, getting everybody connected in a really streamlined way. Any other questions, um, or comments?
Yeah, I mean, I think that one of the, I think like a few of the best things from the retreat were, you know, talking to other people that are doing translational science. As you just said, I mean, like it would have been helpful to talk to Sushma too at the retreat, but I learned so much from chatting with people that are completely outside my field, but doing translational science, because many of the same, you know, resources at Stanford are useful for our different projects. And then, yeah, and the other thing that was just so helpful was the the time to sit and discuss and think and put together a project. And because so often we don't have that, our time is so fragmented between clinical service and sort of administrative tasks. And so it was really invaluable, and we're still executing on those plans that we launched at that retreat.
Awesome. Well, great. Terrific. Well, if there aren't any other questions, just want to thank everybody for their time, Juliet especially, and really appreciate the time you took out just to kind of describe your project. We'd love to connect with folks offline, just more if they have individual questions or concerns or needs. So feel free to reach out to me, only an email away, and I'm just excited to kind of see where this goes from here. But thanks everybody, and hope you have a great rest of the day.
Thank you.
All right.
Thank you.